Formation of notochord. We are going to do the quick revision of notochord. We are going to see about its introduction, its formation, and remnant of notochord in adult human body as this is the embryonic structure and its function. So in this picture, you can see the shape of embryonic disc changes from circular, then oval, and finally become the pear shape. And inside changes are, you can see this is the procaudal plate and this is the primitive streak. So if you look into the introduction of notochord, it is a midline structure that line from the cranial end of primitive streak to the caudal end of procaudal plate. So this is a midline structure and it also replaces the mesoderm and lying between the ectoderm and endoderm. So what are the process of formation of notochord? So you can see in the first change is the formation of primitive node. Primitive node is the thickening of the upper end of the primitive streak is known as primitive node. This is the first step. In the second step, you can see a depression or a pit is created in the primitive node known as primitive pit and blastopore. In the third step, you can see this solid mass or the cells again outgrow towards the cranial end of embryonic disc. This is the cranial end of embryonic disc and this is the caudal end, and it is decided by this structure that is known as procaudal plate. As this structure develops, this is the circular embryonic disc. In we can't decide which part is going to be head and tail, but after the development of caudal plate, we can say this part is going to be head and this part is going to be tail, or we can say cranial and caudal. So this process grows towards the cranial end of embryonic disc, and now this is called notochordal process, and it also replaces the mesoderm in between. This is the ectoderm, mesoderm, and endoderm. It also replaces the mesoderm here. In the fourth step, you can see the primitive pit, or we can say blastopore, also uh, make a cavity alongside the whole of the notochordal process. Now this cavity is called, due to formation of this cavity, this notochordal process now called notochordal canal. So fourth step is the formation of notochordal canal. In the next step, the cells of this notochordal canal replace the, or we can say intercalated with the endodermal cell. There is a mixing of endodermal cells and the cells of the notochordal process. And finally, in the next step, it replaces these cells. As we all know, above this is the amniotic cavity and below this is the yolk sac. So you can see here, through the blastopore, this canal is communicating with this amniotic cavity. And after the intercalation, these cells are replaced by, and again this cavity communicate with the yolk sac. It also remove the cells. So now, like, blastopore is communicating with the amniotic cavity, and at the lower end it communicating with the yolk sac. So a canal is created from amniotic cavity to the yolk sac. That canal is known as neuroenteric canal. Neuroenteric canal, very important structure. So in this picture, you can see the neuroenteric canal. This is the amniotic cavity, yolk sac cavity, ectoderm, mesoderm, and endoderm. And this is the primitive pit, and this is the rest part of this canal that communicate with the yolk sac. The important structure is formed, that is the neuroenteric canal. It is a temporary structure, temporary canal, which later on fused. So why this form? As the name suggests, neuro means related with the nervous system and enteric means related with the digestive system. So for a short span of time, there is a communication between the function of this comes under the function of neuroenteric canal. First is there is a exchange of cells and molecules and signaling between the nervous system and the digestive system, which is very important for the proper development of nervous as well as the digestive system. And later on as this is a temporary canal, later on it fuse. But what happen if it doesn't fuse? It will lead to the formation of neuroenteric cyst that present in the spine and brain also, and it will lead to the other malformation if it unable to fuse in later on. In this picture, you can see the rearrangement of the cells and finally the formation of notochord. In the first picture, you can see here is the solid mass-like structure that is known as notochordal process. It lies between the ectoderm, mesoderm, and this is mesoderm, and this is the endoderm. So it is a solid mass-like structure. In next, you can see a cavity is created. That is now this structure is called notochordal canal. Finally, the intercalation in the embryonic endoderm. Here it is endoderm. And after the intercalation, it become just a communicate with the yolk sac and become a flat uh, uh, plate-like structure. And now this is called notochordal plate. You can see it rearrange itself in the place of endoderm, and finally it again go back in its position, and finally it will lead to the formation of definitive notochord. So in this way you can quickly recall how this notochord form. Now its remnant in adult human body. As notochord lies in the midline, but it does not form a part of a vertebral column, but a part of an intervertebral disc. As you can see in this picture, these are the vertebral bodies and intervertebral disc. And if we look at the superior view, we can see the annulus fibrosis here and the nucleus pulposus in the center. This is jelly-like structure will help in bearing the pressure as well as act as a shock absorber. So this is the remnant of the notochord. This is the remnant of notochord. The and second remnant of notochord is the apical ligament of dense of axis vertebra, which is two vertebra. So these are the two remnant in adult human body, which represent the formation of notochord during embryonic life. Next, what is its function? The function of notochord. First, it acts as a inducer, or we can say secondary inducer for the formation of neural tube. Act as a secondary inducer for the formation of neural tube, and it is also required for the as we can see in form of a neuroenteric canal. It is required for the proper signaling and exchange of molecule in form of a neuroenteric canal, and mainly it is act as a secondary inducer for the formation of neural tube, and primitive streak act as a primary inducer for the formation of notochord and the intraembryonic mesoderm, and after the formation of notochord, formation of neural tube takes place. So this is a quick revision about how giving the introduction and its formation and what are the remnants of notochord